Hello everyone. Today we are going to begin with part 2 of molecular basis of inheritance. Now in the first part we have already gone through some experimental contribution given by scientists. The contribution that they gave was which nucleic acid is acting as a genetic material, right? Today we will start with a lecture of comparing between a DNA and an RNA. Now we all know that DNA is present in most of the organisms, right? While RNA is present in some viruses. Whereas we know that DNA is double-stranded and RNA is single-stranded, right? Then we also know some basic, now when I am saying we know means what? We have already gone through these concepts earlier, right? That DNA has what nitrogenous bases? That is A, T, G, C. That is adenine, guanine, thymine, cytosine, right? Adenine, guanine, thymine, cytosine, A, G, T, C, according to what I said, right? And if I have to go with RNA, RNA has nitrogenous bases like what? A, U, G, C. Now, instead of thymine, which is present in DNA, the RNA has what here? Uracil. Okay, so they have adenine, uracil, then cytosine and guanine. They do not have thymine. So, instead of thymine, there is a substitute which is known as uracil. Let's go into the detail about DNA and RNA comparison. The DNA is double-stranded, right? With exception of some viruses, whereas RNA is generally single-stranded. Now, we all know that DNA is a genetic material which is present in all the living organisms, while RNA is present in only some viruses. Okay? Now, which is the sugar which is acting in DNA? We all know the full form of DNA, right? That is deoxyribonucleic acid. So which sugar is acting here? It is deoxyribose. Okay, whereas in RNA we know it is ribonucleic acid. So the RNA, the sugar which has is what? Ribose. Correct? So the nitrogenous bases present in DNA is which one? Adenine, guanine, thymine, cytosine. Whereas in RNA the nitrogenous bases present are adenine, guanine, cytosine and uracil. Now, DNA can be less reactive compared to RNA, right? Now, why are they less reactive? Because they are chemically and structurally more stable. Whereas RNA, they are chemically and structurally less stable. Okay? So, this is the comparison of DNA and RNA. Let's begin with the next topic, that is packaging of DNA helix. Now, here we are going to learn about in eukaryotes as well as in prokaryotes. Now, let's begin with the first one, that is eukaryotes. Now, how is the packaging here? The organization of the DNA is more complex as compared to the prokaryotes. So, depending upon the amino acid residue, a protein is acquired, meaning the protein acquires its charge. So, the protein which is present here is known as histones. Okay, what is it known as? Histones. Now, this particular histone, which is a protein, is actually basically rich in an amino acid, which is known as lysine and arginine. The two names of the amino acid are lysine and arginine. So both of these amino acids carry positive charges on their side chain. So histone is said to be what? It is a set of a positively charged protein, right? So when these histones organize themselves as a unit of 8 molecules, this is known as what? Histone octamer. So a DNA has been wrapped. The DNA, when I say DNA is what? A negatively charged, right? So the negatively charged DNA is wrapped around this positively charged histone octamer. This forms a structure which is known as a nucleosome. Fine. Now this histone proteins are of 5 types basically. Now these are what? The H1, H2A, H2B, H3 and H4. Okay. One nucleosome contains approximately 200 base pairs of DNA helix. Now, this nucleosomes are the repeating units of chromatin, which almost looks like beads on a string when you see it under the electron microscope. Fine. Now, I have to talk about the next, that is NHC protein, which is known as non-histone chromosomal protein. Now, these are additional sets of proteins that contribute to the packaging of chromatin at a higher level. So these are basically of 2 types. One is euchromatin and one is heterochromatin. Now this euchromatin is a region of the chromatin which is lightly stained. Okay, meaning it is loosely packed in a typical nucleus. Whereas the heterochromatin which is there, it is densely packed, or you can say darkly stained, right? This was about the NH3 protein that we need to know. Then comes the main topic, that is central dogma. Now, the central dogma covers what? The molecular biology, which states there's only one way or unidirectional flow of information from master copy to the working copy. Now, if I have to give like what the master copy DNA to the working copying RNA, you would term it as what? Transcription. And from working copying RNA to building plan polypeptide, that is known as translation. So, how does it go according to the diagram shown? It is the DNA to mRNA, mRNA to polypeptide. How is that DNA to mRNA? You would say it as transcription, right? And if it is DNA to DNA, that is what? Replication, right? Then mRNA to polypeptide is what? Translation, right? So this is the central dogma. But in transcription or RNA synthesis occurs over the DNA, translation or protein synthesis occurs over the ribosomes. Fine. Now in the eukaryotes, the two are separated in time and space. This protects DNA from respiratory enzymes like RNAs and nucleases. Okay. So if I have to talk about the replication of the DNA, in addition to the double helical structure of DNA proposed by Watson and Crick, a scheme for DNA replication. So according to this model, the two strands of double helix would separate and act as a template for the synthesis of new complementary strand. So the template strand is a complementary strand. After the completion of the replication, each DNA molecule, right, would have one parental and one newly synthesized strand. So what is it happening? Replication. It is forming its new strand. So this scheme for DNA replication was termed as what? The semi-conservative DNA replication. Okay. Now this was actually experimentally proved, the replication of DNA, by the two scientists whose name is Messelson and Stahl. The complete name of Messelson is Matthew Messelson. And the complete name of Stahl is Franklin Stahl. In which year? 1958. Now they conducted an experiment based on what? E. coli. The name of the bacteria, which is E. coli, that is Escherichia coli, to prove that DNA replicates semi-conservative. Now how did they prove? Now they grew many generations of E. coli. Now they grew it in a medium which contained the heavy isotope of nitrogen as the only source of nitrogen. The result was that the heavy isotope was incorporated into the newly synthesized DNA by centrifugation in a cesium chloride solution, density gradient. This heavy DNA molecule could be distinguished from the normal DNA. So the cells were then transferred into a medium containing normal NH4Cl, right? So the samples were separated independently on the cesium chloride gradient to measure the density of DNA. So at definite time intervals, as the cells multiplied, the samples were taken and the DNA which remained as double-stranded helices was extracted. So the DNA obtained from the culture one generation after the transfer from 15N to 14N medium, that is after 20 minutes, say like 20 minutes, because E. coli divides in 20 minutes, that is the reason. I had a hybrid, okay. So the DNA obtained from the culture after another generation, you can name it as a second generation. It was composed of equal amounts of hybrid DNA and the light DNA. Fine. So similar experiments were on beans, which is known as faba beans, were conducted by Taylor, okay, involving use of radioactive thymidine. Fine. The results were that the DNA and chromosomes also replicate semi-conservatively. So now there are certain enzymes which are involved in DNA replication. Let's move ahead with this. Now, for the process of replication, you require a catalyst, which is known as an enzyme. So, which in all catalyst you require? One of the name of the enzyme is DNA-dependent DNA polymerase. Okay. Now, DNA-dependent DNA polymerase, it is one of the main enzyme which is used as which uses the DNA template to catalyze the polymerization of deoxynucleotides. Okay. So in prokaryotes, there are three types of DNA polymerases. One is DNA polymerase 1, then 2, 3. Whereas in eukaryotes, 5 different DNA polymerases have been identified. What are these? This is alpha, beta, gamma, delta and epsilon. Fine. Now these enzymes are quite efficient and they can have as they have to catalyze polymerization in a large number. Okay, so therefore these enzymes also help in removing mismatched nucleotides by a mechanism which is known as proofreading. Now the second enzyme which is involved in DNA replication, or which is important for the DNA replication, the name is helicase. Now what does a helicase do? It unwinds the DNA strand, that is separates the two strands from the one point for the formation of the replication fork. Okay, so the third enzyme is topoisomerase. The unwinding of the DNA creates a tension in the DNA strands, so which get released by an enzyme which is known as topoisomerase. The last one is DNA ligase. So it facilitates the joining of DNA strands together by catalyzing the formation of a bond, that is phosphodiester bond. So it plays a role in what? Repairing the single strand breaks in duplex DNA. Okay. So we got to learn about all the four enzymes which is important for DNA replication, that is DNA dependent DNA polymerase, helicase, topoisomerase and DNA ligase. Right. So let's move ahead with the next important topic, that is process of DNA replication. Now the replication is an energy expensive process. Now deoxyribonucleoside triphosphate, that is DNTPs. Okay, what is it? Deoxyribonucleoside triphosphates, that is DNTPs, serve the dual purpose. One, acting as a substrate, another provides energy. Okay, so in a long DNA molecule, replication takes place within a small opening of the DNA helix, which is known as replication fork. Okay, now DNA dependent DNA polymerases, it catalyzes polymerization only in one direction. What is that? 5' to 3' end. Now this brings additional complication at the replication fork. Now due to this one strand, that is template with polarity 3' to 5', replication is continuous. So this DNA strand is called leading strand. Okay, what is it known as? Leading strand, since the replication is continuous. While on the 5' to 3' strand, the replication is discontinuous, that is in small fragments, which is called Okazaki fragments, thereby forming lagging strands. These fragments synthesized are later joined by the enzyme which is known as DNA ligase. Okay. Then comes the DNA polymerases which cannot initiate replication process on their own. So replication does not initiate anywhere in DNA. So it begins in the definite regions in a bacteria, that is E. coli DNA molecule. So that beginning is known as what that particular region, you would say it as what origin of replication. Alright, which has recognition site for DNA polymerase and which also provides site for attachment of RNA primer. Okay, next we need to learn is about the transcription. Now what is transcription? Transcription is a process of copying the genetic information. Okay, it is a process of copying the genetic information from one strand of the DNA into RNA, as we have learned in the central dogma, right? From DNA to RNA. So meaning what? The copying of genetic information is from one strand of DNA into RNA. So also the process of transcription is governed by the principle of complementarity, except the adenosine now forms base pair with uracil instead of thymine, right? So unlike in replication, which once set in, the total DNA of an organism gets duplicated in transcription. So only one segment of DNA or only one of the two strands is copied into RNA. So the transcription unit of DNA is defined as what? The primarily three regions. Now what are the transcription unit basically composed of? It is a promoter, a structural gene and a terminator. Now a promoter, structural gene and a terminator, we will go to it in detail, but let's learn what is it. The two strands of DNA which is there, it has opposite polarity, right? 5' to 3', 3' to 5'. And the enzyme DNA dependent RNA polymerase catalyzes the polymerization in only one direction, that is 5' to 3'. So the strand with 3' to 5' polarity acts as a template and is known as a template strand. And the strand with 5' to 3' polarity and same sequence as RNA except thymine at the place of uracil is displaced during the transcription and it is known as a coding strand. as you can observe the schematic representation of a structure of transcription unit there. So a promoter, structural gene, and a terminator. So a promoter is what? It is a DNA sequence. What does it provide? It provides binding site for RNA polymerase. And where is it located? In the 5' end, that is upstream. Okay, in the 5' end it is located of the structural gene. Fine. And its presence also defines a template and coding strands. While the structural gene in the transcription unit is flanked by the promoter and the terminator. While terminator is located towards the 3' end, which is a downstream, okay, of the coding strand. So it usually defines the end of transcription process. Fine. Now this transcription unit and the gene is a functional unit of inheritance. So the DNA sequence coding for tRNA or rRNA, that is transfer RNA or ribosomal RNA molecule, also defines the gene. So here what is a cistron? Cistron is a segment of DNA that codes for a polypeptide. So there can be monocistronic or polycistronic. Now if you have to talk about in case of structural gene, it can be monocistronic or polycistronic. So monocistronic is present mostly in eukaryotes. Okay, and polycistronic is present mostly in prokaryotes. Fine. Now these monocistronic structural genes which is present in eukaryotes have coding sequences. One is exon and one is introns. Now exons are the coding sequences or expressed sequences that appear in mature RNA, while introns do not appear in mature RNA. Now if I have to go in detail about types of RNA and the process of transcription, types of RNA, we know there are 3 types of RNA which is found in bacteria. Okay. Now mRNA, one is mRNA, one is tRNA, and one is rRNA. mRNA which is a messenger RNA, tRNA which is a transfer RNA, and rRNA which is a ribosomal RNA. Now mRNA, what does it do? It provides the template for translation, and tRNA which brings amino acid and reads the genetic code. Well, rRNA, what does it do? It plays structural and catalytic role during translation. So all the 3 RNA basically are needed for what? Synthesis of protein in the cell. So I hope you understood the concept which is being taught today. Kindly go through it properly, study well. Thank you so much.